Welcome to this latest webinar with the Product Quality Cleaning Workshops team. And this one is a little different. We're going to be introducing the Cleaning Research Group. And so today we're going to answer that question. What is the Cleaning Research Group? Of course, this is brought to you by the Cleaning Workshops team, Barbara and Ed Kanegsberg, who are the cleaning lady and the rocket scientist. And they've been doing critical consultants and critical cleaning for many years, authors of the 2-volume CRC handbook on critical cleaning. As you can see here, it's still a great resource. I go to it frequently. And you can find them at BFK Solutions, and there's their emails.
Right, and here's Darren Williams, the professor, and he actually teaches thermodynamics in a way that even I can understand some of it, which is quite wonderful. He is at Sam Houston State University. He is the leader of the PAC for the Cleaning Research Group, and he co-chairs with us the Product Quality Cleaning Workshops. He will also do cleaning trials, and as we do, he will chase squirrels for you, whatever you need to do. And here's how to find them.
That's right. And we've been chasing lots of squirrels this summer, and that's what we're going to be talking about. So here's our featured guests, the Cleaning Research Group. We have the undergraduate students, Payton Barkman on the left, Hailey there in the middle, and then Zachary. And then Tommy over here on the right. We're kind of covering him up with our faces. And so this is a little chance for them to tell you about what they've done this summer and some of the things that they've learned. And so let's jump right in.
Great. So these are the vial cleaning tests. You could click next.
Yeah, sure.
I'm sorry. Was I supposed to introduce myself there?
Yeah, that's
first,
okay. Go ahead.
Okay.
Tell them your name.
Because in my brain, we would do that on the screen where we're all there.
Yeah.
It's fine. We can cut this, right? Question mark.
Sure. Go for it.
You're there forever, but keep going.
So my name is Hailey. I'm majoring in chemistry, and I'm looking to work in the cosmetic formulation industry. And so here we were doing our vial cleaning tests. So we did a gravimetric test of 12 different cleaners and 5 different soils. And we were doing this primarily to set up a procedure for the capillary tests that we did after this. So in a quick summary here of what we were doing, we would weigh the vials and then we would contaminate them with the soils. And you can see in that bigger picture there, we are blotting it on some chem wipes so that we don't get excess soil on the outside so that we can only measure what's on the inside being cleaned. So then we would weigh them again, and then we shook them 20 times, rinsed them the same way, and then we would dry it in the GC oven and take the weight again so that we could have a comparison of how much soil was cleaned.
Yeah, pretty simple study. And
that's
it was
how well these cleaning agents are removing soils.
Yeah, and we didn't want to have a really strong, like super effective cleaning method because we wanted to test the chemical cleaning ability of the cleaners. So just 20 shakes in a vial is not very strong cleaning, but these cleaners did really well. They cleaned most of the soils.
They did very
They all did really well. Yeah.
But the soils were easy.
Of one soil.
Yeah.
All of them came off extremely easy, so we needed to. Yeah. It's part of the reason we were doing our capillary tests is that would be a more difficult form of cleaning, hopefully.
Yeah, that's right. So, oh yeah, that's the shaking. Very good.
Yeah, the animation broke a little. And so here's just a closer picture of the before and after. So there's the before where it's contaminated and the after it's been cleaned.
Mm-hmm. And this is, we zoomed in so you can see how difficult it is to image the soil, and we'll see later how this was improved using fluorescent light.
See a little bit at the bottom.
Yeah. Yeah, a little bit. There's some kind of film there. Yeah. Okay, so then we made it more difficult to clean with the capillary cleaning tests.
So I'm Zachary, majoring in chemistry, and here's our capillary cleaning test. So we just started with empty capillaries, measuring them all gravimetrically. So getting the initial, the dirty, and then the cleaned one, those pictures being on the left in order, using our volume change cavitation system, which is shown in the bottom right. And we had to clamp it to the workbench because it's moves quite a bit, as you'll see in the next slide,
Yeah, we
whenever
have a
it's in
video. Yeah.
use.
I didn't catch. Did you say what your plans are for the future? What do you?
Oh, currently it's undecided,
Okay.
but most likely just go in industry.
Okay. Fantastic. Yeah.
Undecided is always fine.
Yeah. And you can see how these the fluorescent light really helps image that soil. So this is a video of that cavitation cleaning device. Okay. There's different versions. I have the industrially like cleaning machines that use this same science. They call it cyclic cavitation, vacuum, like cyclic nucleation process, I think is one of the acronyms. We call it vacuum cavitation because that's the science behind it. So here's a video of it in action. So you can see the cleaning solution in this tube. That tube is actually our cleaning chamber for the capillaries. So we can put the capillaries in that tube and clean them with vacuum cavitation. And so you can see the device moving around. That's why we clamped it to the bench. So just a little short video of the cleaning process. You can do that 20 times and clean the inside of these capillary tubes quite effectively.
What's the inner diameter of the capillaries?
The capillaries, these capillaries that we have, well, Zachary, do you want to take the questions? Do you remember
I do not know the inner dynamic
Okay.
diameter.
The inner diameter is about 1 millimeter. So it's a very narrow opening. And then it's about 100 millimeters deep. So it's a 1 by 100 aspect ratio blind hole. So
Yeah.
And that's real hard to clean.
very hard. So think about if you had a metal part and you drilled a hole this deep, okay, and it had chips and cutting fluid and everything inside, that would be a very difficult hole to clean with an aqueous cleaner. And we can show with this vacuum cycle cavitation that you can get in there and you can suck those chips and oil out, push in cleaner, suck that cleaner out, push in some cleaner and pull that out, and then you can rinse the cleaner out as well.
And in the big machines, they actually do that by varying the pressure to modulate the boiling point a little bit. So you constantly get that first boil.
Yeah, exactly.
And it's real cool for
Yeah.
cleaning hypodermic needles.
Oh, yeah, that's right.
Yeah.
All right.
And then at the end of our capillary tests, we made fluorescent versions of our 5 soils so that we could test the percent removed by the fluorescence analysis using the ImageJ software.
Okay.
And that's what's shown on the right. And on the left was our picture setup. We standardized it so that each picture would be the same, as it shows we have our phone holder there, and then our UV source is that silver block on the left.
So we shine UV light on the fluorescent soils, and then they fluoresce, and then this is holding the camera, this thing right here. So these, cameras on cell phones are really good nowadays. And so all you really need to do is just have a mount for that phone. You can take your images using a cell phone. And then we just use this alternate light source, a UV light source. And these are the images we get. We can see the fluorescence on top, but then the red is actually ImageJ finding those fluorescent pixels and counting them. Notice how this is much easier to quantify than these images.
Yeah.
Right. So there's soil in here, but we really can't identify it. It's difficult. And so using fluorescence is a very good technique to be able to quantify soil residue on a part. Okay. Just talking about the physical properties.
Hi, my name is Payton. I'm majoring in forensic chemistry and I'm aiming for grad school and then a job in a forensics lab as a toxicologist. My favorite thing about the summer's research was making the procedures and Excels for the co-distilling test.
Okay, great.
So the top left and rightmost pictures are the pH calibrators and the pH meter. We took the pHs of the cleaners that we used after calibrating the meter. These would go on to be used as a way to compare the cleaners. The two bottom left pictures are the 4 liquid contaminants after we cooked them and the setup for determining their boiling points. The boiling points of the contaminants were useful for the co-distilling tests that I will discuss further in a moment.
Let's talk about the colors of these contaminants. Were they this color when you started?
They were not. We had 2 contaminants that were red and 2 contaminants that were clear. The red ones are the 2 frontmost ones, and the clear ones were the backmost
Mhm.
ones, which have turned yellow and orange.
Yeah, how about that? So this red one stayed red, but this one was red and kind of turned an orange color. So,
Yes, a bit.
yeah. And that's a good point. We bring up the Barbara and Ed and I whenever we talk about challenging cleaning situations. If you take your soil like these hydraulic fluids that we have right out of the bottle, they're very easy to clean off. But that's not necessarily the state that you're going to experience when you're cleaning a part. If it's been machined or heated up, that soil's been cooked on there or changed in some way. And so it's what we're doing here is a good job of changing these soils in some way. We haven't yet, but we'll probably take FTIR before and after we cook them and see what kind of changes have taken place.
That's important because the residue is not the same as the soil.
That's right. Yeah.
Hard to use it. It's a mess. Yeah.
This
The
was
picture
cool.
on the left is our tensiometer, and the picture on the right is a close-up of it in use. We used it to measure the surface tension of our cleaners. Like
Yeah.
pH, these measurements were used to compare the cleaners. So if you would hit the spacebar, the picture on the right is actually a video of the moment the surface tension of the cleaner was broken.
Yeah. Okay. Let's see. Nope. Let me go
Cannot
back and
play
try that
media.
again. There it goes.
Okay.
That's nice. And so this little ring that is showing is being the mass of that ring plus the liquid hanging off of it is being measured by a balance that's up on top. So that's how we're measuring the surface tension. And this is a little Pyrex sinker that allows us to measure the density. So some physical properties measurements. Okay, this is interesting. What's this?
These pictures were from our co-distilling tests. We plan to extract any remaining contaminants from the pieces we clean with perchloroethylene, also known as perc, evaporate off the perc using the gas chromatogram oven, then add a known amount of perc back into the contaminant to analyze the concentration of soil left. In order to do that, first we have to ensure that the perc does not co-distill the contaminants. These pictures were the final times recorded for 2 of our contaminants, one of which, the one on the left, was put in the GC oven at 122 degrees C, and the other one was left in our fume hood at 18 degrees C, which is about room temperature for our lab.
Yeah, really cold lab.
That's cool.
Yes.
Yeah,
That was on a warm day.
that's right. Yeah, sometimes it's 17 C. Yeah, it's
Oh, yeah.
crazy. Yeah. And so, yeah. So before we go on, I mean, so what we did see was some of the lighter hydraulic fluids did have a low enough boiling point that a substantial amount left with the perc. And we talked
Yes.
about this in one of our recent webinars, Barbara and Ed, about cleaning with some of the new alternative solvents, that if the cleaning solvent has a too high of a boiling point, then it's hard to separate from some of the lighter soils that come off the parts, because the equipment it stays in the solvent when you distill it.
It's a problem.
Yeah.
Hello guys, my name is Tommy and I'm majoring in chemistry as well. And I plan to go to industry. Don't know which one yet, but that is to be determined. But here I wanted to talk about the construction of our cleaning device, the one that you saw earlier in the video. So DW made the first one, and then I got to get hands-on and make the second one that we had. And I wanted to point out some of the things that, some problems that we had with it whenever I was making it. So in between each section here, there's a bunch of different pieces that come together to make this. We have a 3-way that connects to our valves, and then those valves connect to our lure locks, so we can hook up our syringes. And in between each of these pieces, we use Teflon tape to ensure that we didn't have any leaks, since we're going to be pulling a vacuum on this. And what I didn't notice, or didn't think I was going to have an issue with, was I had a leak right at one of the valves. So that red circle there shows where I had one of the leaks, and I never thought I would have that. You know, I thought it would come ready and leak proof.
Okay.
But that was an easy fix. All I had to do was tighten up one of the nuts that is on the handle there, and all the leaks went away, and it was good to use, and it worked just fine. We used it for many different cleanings. But also, this kind of shows too the cap that you see laying there at the top. That's kind of what we cap on before we start cleaning. So we're able to add the capillaries through the top there, and then cap it, and then start a cleaning process. So, and then to show you the clamp too, that we had to clamp the whole cleaning device to the bench, so it didn't move around too much when we're pulling on the syringe. So
Yeah. And again, these types of syringes were okay for the aqueous cleaning, but when we go to our rinsing with PERC, we're going to have to get rid of all of the polymers because.
oh yeah, you'll see that later. That's something that we'll kind of talk
Oh,
about.
yeah.
Yeah.
I've spent a lot of years analyzing polymers instead of what I thought I was doing. Yeah.
Here also, I was the one kind of doing all the 3D printing. So if we ever needed something in the lab to work a certain way, then I would get on Fusion 360, design something. And I know they don't look pretty, but they definitely work. So this first one here is for our FTIR. We have a quart cell. We have many different varying sizes of quart cells, and the longest one here is 10 centimeters. And so we wanted to make sure that we had something that could hold that 10 centimeter quart cell and let the beam go all the way through. And then the second one we have here, which is one that you already saw, it was for the fluorescent test to hold your phone. I wanted to make sure it was something that we could slide back and forth. It was very easy to use. You could just lay your phone on it. As long as your phone has a flat back end, it'll work with this. And it was very easy to slide it back and forth. So if you needed to move your phone out of the way, grab your capillaries, slide it back, it was very easy to use. This
Again,
last
so
one,
this is designed in the lab and then printed. How many days did it take to sort of design and print this?
it really only takes me a couple hours. First, I need to go to the lab and get my measurements with a caliper, and then I just go on Fusion 360. I've gotten pretty good at using it, so it doesn't take me very long. The printing process is what takes the longest. So
Yeah.
I think the first piece there took an hour, but the second piece, the phone holder, was like 3 hours.
Yeah. So like
So
this first piece is about an hour for printing. Let's say, let's be generous, an hour for design. So you can have this part in 2 hours. That's faster than Amazon.
Yes.
true.
Because you can buy this part. I mean, you can go out there, you can go to International Crystal Laboratories or someplace like that, and buy a holder for a 10 centimeter quart cell. But by the time you go through the purchasing process, and then they've got to ship it to you, and then you got to receive it, and all this. I mean, it takes. I mean, this is even faster than next day shipping because this is 2 hour,
Yeah.
2 hour
Yeah.
shipping.
I think another like long process is when we have supports on these, we use PVA, so we can just dissolve it in water. It's pretty easy
Yeah.
to use. That first one didn't really need many supports,
Yeah.
but the second one did. So
Okay.
another process would be putting it in water, letting the supports dissolve away.
Okay.
So that added to some time too. But
So we don't show it, but the 3D printer we have is an Ultimaker 5S, and it has two extrusion nozzles, so we can run the polymer that we want to make it out of. Was this out of
tough PLA.
PLA, and then we have the polyvinyl alcohol. There's a second polymer that's water soluble. So you print the part, and the water soluble part helps hold up the arches, like on these little circles, and then you can print a really well precisioned part and then dissolve away the support material. What about this
Yes.
last one, this fifth one?
So that last one, we were trying to hook up a vial to our cleaning system, and the vial would essentially be our cleaning reservoir.
Yeah,
And
so in place of this tube here, the vial would
Yes.
go right here. So we needed a 1/4 inch outer diameter piece that would fit into a vial cap. Okay.
So with printing with this filament, we found out that it was airtight, strong enough. I could just make a 1/4 inch tube essentially with this filament. And then at the bottom there, I plan to use that inside of a vial cap and epoxy it together so it's airtight. And essentially you could just twist the cap onto a vial and it should work, right?
Yeah, it should.
but then you go to the next part. So
Yeah.
this is us trying it out for the first time. This was
So
yesterday,
you
actually.
see that little tube, that tube right there was 3D
Yes.
printed, and it's like a little top hat. So the brim of the top hat is on the, inside the lid of the vial, and we had a little trouble getting the vial to close all the way. So you see,
So you see all that water spurting out. So that's not, airtight. It's not good. But, we can work on that.
yeah. So that's again, version 1. We go to version 2 and
Yeah.
make it better. But that's part
Always.
of the learning process. Yeah.
Always. I will say with all my prints too, the first print is never perfect. It's never good. So even that first print there for the FTIR, my first print, the V was too low. So I had to, pick up the V a little bit. And
Yeah.
for the phone holder, the circles, they weren't thick enough for to be very stable. They were breaking
Yeah.
apart. So, I mean,
That's a good point too. So it's not like it's 2 hours of time and it's guaranteed to work. Sometimes the printer needs babysitting and it will screw up, and other times you got to do a Model 2 or Model 3 on the design. Yeah.
exactly. Yeah. But that's, the big part of science,
Yeah.
First thing isn't always going to work, but as long as you know how to fix it, you're good.
Mhm.
So.
And beyond Amazon delivery times, this is a really important skill to have, and I think all of us
Yes.
need to have it, because you need sometimes we need custom things for fixturing for unusual parts, and as we get into tiny parts, they don't make miniature fixtures really well, as I have discovered. So 3D printing custom stuff can be really good. So
Yeah, I will say filament, for us at least, I haven't been able to get super low, like 1 millimeter sized prints or anything like that. That's when it starts to kind of break away, not print very well. But
yeah,
I know there's other 3D printers you can get
there are.
resin or something like that. That would really help.
Sure.
So
Sure, you can get things for itty bitty parts that, yeah.
Yeah.
so this last piece here, this was the most interesting to me. So we were trying to get an IR spectrum of our perc, and we kept seeing just hydrocarbon contamination, and we're like, man, why is this here?
Wow.
So we would distill our perc, and so all the hydrocarbon should be gone, right? Well, we were still seeing it. And one weekend I left one of the syringes that I had full of perc, and I came back and I saw the plunger there started swelling up. I was like, whoa! So that's where our hydrocarbon contamination is coming from. So my big takeaway from this was you really need to be careful about what solvents you're using, what they react with, whenever you carry it over to certain things. So, but at the top here, the top right, that's our IR cell that we put into our FTIR. And I have at the bottom here something that I thought was so much fun for all of us. It was a really good day where we got to clean the salt windows, or not clean them, but polish them so they weren't so foggy anymore, because the humidity here in Huntsville isn't nice to our salt windows. So, but that was one of our, I think, most fun days. So I had to put it in here. So
Yeah, that's good.
Okay.
All right. So, of course, for the video folks, please put your questions in the comment section if you have them, and I'll get back to you with those. But Barbara and Ed, what kind of questions do you have?
Oh, I'm going to ask to anybody one that I asked at the start, right before he started this. To anybody, what do you think you're going to do to change the world? What would you like to do to change the world if you could in your next career? Come on.
I know one big thing that I've always had my eye on, and it's something that I started thinking about whenever I was in PQCW when we were talking about energy. Solar cells. If I could figure out that to get a better conversion from solar energy to electrical energy, I would love to do that. But obviously I don't know all the science behind that, but that would be something so cool to say that I was a part of that. And I know that would be a very big win in the energy department. So
Very cool. Very, very cool.
And with that, even a very, very small increase can be very profitable.
Oh, yeah.
I know. Because what right now is it at 30-40%?
I don't know if it's that high yet. Yeah.
Oh,
but it's
really?
getting,
Okay.
maybe the research cells, maybe they, yeah.
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 I've got another question. Is anybody interested in going into teaching?
Never thought about it. I did have a lot of interest.
Okay. Yeah, Haley. Yeah.
Want to talk about it at all? Or?
I would love to. I currently I've been a teaching assistant for about 3 years now, and I love it. I really do. Getting to help someone understand something that I love so much is just really motivating. Um,
Sure. Sure. Do you want to teach little kids, big kids? Anybody want to teach practicing scientists and engineers? And have you figured out what level yet? Or
it's not my current main plan, but if I were to go into teaching, I would probably want to work with kids in high school,
Sure.
because that's a lot of where they base what they want their futures to be. And if I could convince some of them that science is in fact very cool.
Cool. Okay. Anyone want to discuss what they think might be their most challenging roadblock and how they plan to overcome it? Nobody has any roadblocks.
Roadblock in the sense of this set of research, or as in
in terms
toward our
of
future?
how you might reach your goal. If you're going to graduate school, if you're going into industry, what do you think, or what do you think is a challenge as a younger person entering the workforce?
I think getting my foot in the door. That's kind of why I'm in grad school right now. I wanted to make sure that I stood out a little bit.
That's
And my past job experience too, that's something I can talk about a whole lot. And I think that'll really help me stand out from other people. So I've always been trying to do something new. You can ask DW, even in the lab, like when we're trying to fix the FTIR, like I want to know something new so I can say, hey, I've done this before, you know, I can be a good help.
Yeah, we had the laser module go out on the FTIR, and so it took several weeks for the parts to come in, and they came in. Let's see it came in on Monday or Tuesday, and he told me, he said, please wait until Wednesday afternoon when I can help out because I want to see the switch over. So I made him turn all the screws. So that was really good.
But I don't know, just little things like that
Yeah.
Sure.
you don't get in your undergrad.
No.
Just makes you stand out. So those are things that I try to get to.
Speaking of standing out, when this video goes up on YouTube, we're going to put everybody's LinkedIn profiles down in the description. So if you want to know more or follow along with any of the Cleaning Research Group students or myself or Barbara and Ed, you can find our LinkedIn profiles in the description. And we'd love to connect with you on LinkedIn.
So I keep having questions, but I have one more question. DW meant for Darren Williams, for those of you who don't know, DW mentioned that a number of you were surprised at how to learn about cleaning as a separate field, because I certainly was. I surprised myself. I didn't know about it. Does anybody want to comment about anything surprising or?
I was quite surprised at how many different varieties of cleaning can be done. So here we're working on aqueous cleaners, but there are a lot of other forms of cleaners that I didn't know about. And I was also kind of surprised as to how relevant it would be to me going into cosmetics, is
Sure.
looking at different surfactants are really important for makeup removers.
Sure. They're the micelle water that they have everywhere. It's just a surfactant, or and absolutely.
Zachary, it looked like you were going to say something.
Well, I really had no idea the field of cleaning research existed. So when joining the research group, I really had no idea what to expect, and then learned a whole lot this summer.
Yeah.
So.
Yeah, I think that was the same for me too. Whenever I was getting into chemistry, you're never really told, I guess, that cleaning chemistry is a whole big thing, and you don't really see it until you get into it. I think y'all's seminars that y'all put together are really good for people that are trying to learn everything. So we actually watched one of your seminars, the last one that y'all had, and one of the things that stood out to me was when y'all were going through the schematic, and then we actually got to see inside of the big cleaning machine. That was so cool to me. I don't know. I'm really big on machines and schematics and stuff. So being
Yes,
able to see it all work and put together.
it really is a big deal. And if you do analytical work, there's a lot of cleaning involved in extraction.
Hmm.
And
Yeah, I think Payton can speak to that in terms of her, goal of being in a forensics lab. I bet this experience is going to help you.
I know,
Absolutely.
yeah,
Especially when we get in the fall, now that the FTIR is repaired and we're going to start making up our standard quantitative analysis procedures using IR. I mean, that's going to be totally applicable to trace analysis. So you'll
Oh.
be well prepared for that.
yes. You will be a guest star on NCIS in the coming year.
That's right. It'll be wonderful. Yeah.
Sure. I also think that what you're learning has a lot of applicability across the board. When I was involved in guidance systems for cleaning guidance systems for the military, I noticed that some of the lubricants that were used in the guidance systems were the same as what Estee Lauder and Clinique use. So I spent a bit of time trying to get some information from Estee and Clinique. It was a bit of a challenge.
Yes.
Yeah.
Fortunately bound in the military, but it's really, there's a lot of crossover. There really is a lot of crossover.
No, that's really good to hear, honestly. Just because I don't know exactly where I want to go, and
That's okay.
anything I can take from here and move on, it'll just make the transition into a new job so much easier. So
I think
I mean,
it's
that's
great.
the great thing about cleaning chemistry in general is regardless of what field, I mean, we all have different fields we want to go into, but for all of us, cleaning is essential for what we're doing. So it's so versatile for all of us.
Yeah.
Yeah, that's why I was so excited to sort of stumble upon this niche for our university. I was at a cleaning conference in 2010, just presenting on some contact angle work that I had done with my students. And at the luncheon they went around the tables and everybody just said their name and what company they were from. And there wasn't any other university there. It was DuPont and Dow and Boeing and all of these really well-known named companies. And I was looking around going, oh my gosh, I'm the only university in this sandbox.
Yeah.
So I just decided to start the cleaning research group, and the students have been very sought after. And so I'm excited about what's going to happen in you guys in your future. So
Yes, I'm anxious to hear also. It's really great to get to know all of you.
Right.
I
We even follow up with some of your predecessors who have been through the research group and are
yeah, that's right.
now working several years in
Yeah,
industry, and we keep hearing about them.
it's cool.
So that's been pretty good. I thank y'all for being willing to come on and sort of talk about the summer and your research experiences and so on. We'll have your contact information down in the description. And for those of you who are have stumbled across the Product Quality Cleaning Workshop YouTube channel, please like and subscribe and hit that bell so you'll be notified whenever there's a new video. We don't have a common posting schedule, so you definitely want that bell because things pop up pretty randomly. You can always contact me at williams@shsu.edu, and of course Barbara and Ed are at BFK Solutions. And also, if you're looking for cleaning information, go to bfksolutions.com and look at their Clean Source newsletter. You want to take a moment to tell them about that, Barb or Ed?
Oh, oh golly, just a minute. Sure. We did it. We started it about 20-ish years ago as a periodic effort to let people know what we were doing. Self-interest. We've got our own company, and we've discovered that it's become quite a teaching tool. Every month we discuss something, whatever we want. It might be medical, it might be aerospace,
Yeah.
It might be how to rinse. It might be something even more technical with the boundary layer we've covered.
Hmm.
And we cover things like what is happening in the regulatory world as far as we know it at that time.
That's right. Big news released this week on 1-bromopropane or
Yeah,
N-propyl bromide. So
that's right.
definitely want to go see with the latest information. So go to
Abs-
BFK Solutions and look up their newsletter. All right. Well, for the Cleaning Research Group and Barbara and Ed and the PQCW team, we'll sign off. And y'all have a great rest of your day.
Yes, you too. Take care.